Mendel also created what he calls one of his principles, which is principle of segregation. And we're going to go back to what we learned earlier, which was meiosis. When meiosis occurs, gamete cells are made. And during the formation of gamete cells, as I taught you earlier, these cells have half of the information. So basically what's going to happen is alleles are going to separate during meiosis, and only one allele gets transferred into each cell. So here's an example of a parent cell, and these are their gamete cells, which occur after meiosis. And if you notice, only one allele is transferred. These will come back together during fertilization. So this is an example of gametes coming together during fertilization. And then again, if meiosis occurs once that cell becomes an adult cell, you'll see only one allele in each cell. So it's half of the information. The next principle is principle of independent assortment. So when alleles separate during meiosis and the formation of gamete cells, they are going to do this independently of other alleles. So if you have 20 alleles in one cell, and one gamete cell can have all dominant alleles, or one gamete cell can have all recessive. There is no correct path. It's independent, and they do this at random. Again, think of genetics as random and chance. So here's a scenario. Here is a cell, and this cell is going to go through meiosis, and alleles, remember, you're going to get one of each. So one gamete cell can have capital R and capital Y. Another gamete cell can have capital R, little y, and so on and so on. These cells can mature, obviously, and this could be one parent or a set of parents. And if they put them together, so if you put two traits together and you use hybrids, it's called a dihybrid cross. And these are the possible gamete cells for each hybrid, let's say, or each parent. And this would be the father, this would be the mother, and the offspring. In this case, you bring the R down and the R across, which is RR, and then you bring the next allele, capital Y, and that is the genotype for this offspring. If you come here, you're going to bring lowercase r down, uppercase R across, and it doesn't matter, they put the uppercase first. A lot of people like to put the uppercase. It doesn't matter if you put lowercase first. Capital Y, and so on and so on. And I'll just reveal the chart as I did in class so you could see the rest of the information. You will be given a chart like this on the test, and you will have to plug in the information as we did together in class. So sex-linked traits are traits where the gene is found on the X or Y chromosome. 99% of the time, it's actually found on the X. So I'm going to focus on that in this class. Just like any trait, I have to give you a scenario first. So in this case, I'm giving you this scenario, which is baldness. And I'm letting you know it's a recessive sex-linked trait. So this is the name of the trait, and this is the type of trait, recessive and sex-linked. So the first thing you want to do, just like in the other scenario, is figure out what letters to use. Because baldness is actually a disorder or like a defect, we want to say that having hair is normal. And that would be the dominant. Normal would be dominant because baldness is recessive. So we're going to choose always the capital letter of the dominant trait. So if you're normal, you would have a capital N, and if you're bald, you would have a little n. Refresher, females are XX and males are XY. So the possible genotypes for all females carrying a sex-linked recessive trait are as follows. If you have X little n, you're actually bald. X little n, X capital N, you are normal because the dominant masks the recessive, but you are considered a carrier. X capital N, you are completely normal. Male genotypes now, which are XY. Males, pretty much, they carry only one, and they will have the trait because they do not have another X that would mask the recessive trait. So carrying one will give them the disorder. So one gene and they will be bald. And then the only way they could be normal is if they do not carry the gene. So we're going to do a cross of a female carrier with a bald man. So the mother would be X little n, X capital N. The father would be X little n, Y. I'll put the mother on the top and the father on the side. So the difference is now you have to include the sex of the parents because the trait is carried on a sex chromosome, in this case the X. So the offspring, you would have X little n, X little n. So this male would have it. X capital N, X little n. I'm sorry, this female would have it. I'm sorry. In this case, this female would be a carrier. This would be the male probability, X little ny, this male would have it, and this male would be fine. So if you're going to do percentages, you would do it as males and females now. So you would say 50% chance that females will have it, and 50% chance that males will have it. If they ask you questions like, what's the percent chance of a child having it? It would be the same, 50% chance, because you have a male child chance, so 50% chance. If we move on to the next example, where, and this is hypothetical, Harry is dominant and it's a sex-linked trait. So I'm giving you a dominant sex-linked trait. If we're going to think of what letter to use, we're going to use H because H is the dominant trait. So capital H for dominant and little h for recessive. These are the possible genotypes for the females, and these are the possible genotypes for the males. Let's cross a normal male with, a hairy male, because it's a dominant trait, with a normal female or non-hairy. The first probability for females is that they will have it. The second is that they will have it. The male will be normal in both cases. So 100% chance of having a non-hairy male and 100% chance of the female